Hello students, today I am bringing the solutions of ANTS paper 11 botany portions. So let's start with question number 101, okay. So the question is given on water movement, water potential. 3 cells are given X, Y and Z. Values are given. In the X cell, solute potential minus 10 and pressure potential is 5 bar. So in the X cell, the value of water potential will be how much? Minus 10 plus 5, that is minus 5 bar. In the Y cell, another cell, solute potential minus 15, pressure potential 10. So water potential will be how much? Again it is same value, minus 5 bar. Another cell Z, what will be the value of water potential? This is minus 7 plus 1, that is minus 6 bar. So you know that movement of water, it occurs always from higher water potential area to lower water potential area. So here the high water potential value, it is X and Y as compared to Z. In these two cells, same value. So movement of water will be from X to Y or Y to X, it will be same value. And in this Z, water potential is less. So it will draw water from both X and Y cell. So according to the options, you can see water can flow from X to Y, yes, but it is not from Z to Y. So A option, it cannot be correct. B, second option, yes, it's shown X, Y having same water potential value. So water can move in both the direction, and Z cell, it can take water from both the X as well as Y cell. So the correct option for 101, it is second option B. Got it? You can also check the other options, but these are wrong. Clearly it's shown. So let's move to the next questions. 102. One of the following can fix atmospheric nitrogen in specialized cells called heterocyst. So you all be knowing this clearly. Heterocyst, it is this specialized cell where atmospheric nitrogen can be fixed, and atmospheric nitrogen, it is fixed by filamentous cyanobacteria which possess this specialized cell. So let's see. Let's say it is the filamentous. Hetero means what? Different. Okay, this cell is different from the others. So this is the filament of a cyanobacteria which can cause a fixation of atmospheric nitrogen. So this cell, it is different from other, and this is what we call heterocyst, where atmospheric nitrogen can be fixed into one of the usable form, that is ammonia, right? So what is the question? Which can fix atmospheric nitrogen? Agaricus, you know, it's a fungi. It does not have the ability for nitrogen fixation. Second, polygonum, it is an angiosperm, fine. It also cannot fix atmospheric nitrogen. Penicillium, again it's a fungi, right? So very easy. It is what? The last option, that is nostoc. You know students, it is the filamentous cyanobacteria, blue-green algae, okay, which can fix atmospheric nitrogen either in the asymbiotic form or symbiotic form. Okay, it will come in both the category free living as well as symbiotic, right? So hope you did it correctly. Now let's see next question 103. Select incorrectly matched pair. Incorrect. Catalytic RNA, soluble RNA. So what is catalytic RNA? RNA is catalytic. This is one of the reasons how it is less stable than DNA. Catalytic RNA, it is ribosomal RNA, right? Ribosomal RNA. In case of prokaryotic cell, you studied that in case of bacteria, the catalytic rRNA is which one? In case of prokaryote, the catalytic rRNA is 23S, ribosomal RNA, right? Present in 50S, larger subunit of ribosome. Then in the eukaryote, the catalytic RNA is 28S, which is again present in larger subunit of ribosome, that is 60S. Fine. So what is the given combination? Soluble RNA. No, it cannot be correct. Okay, soluble RNA is the smallest RNA, transfer RNA, adapter RNA we say. So other options also we will judge. Second option given SNRP, okay, which is small nuclear RNA. So this is, it is correct, SNRP, which is utilized in second option SNRPs, and it is small nuclear ribonucleic acid. It is correct. As you know, you studied in post-transcriptional changes. So in the last step, that is in splicing, this SNRP is utilized. SNRP is utilized for what? Removal of non-coding sequences of primary transcript, that is intron. Okay, it is utilized for splicing. In case of eukaryote, for post-transcriptional modification of primary transcript, right? So this is a correct combination, correct match. Then capping guanosyl transferase. So this is also correct, capping. Okay, capping means addition of 7-methyl guanosine triphosphate, okay, at the 5' end of primary transcript, 5' end of primary transcript. So this is the first step of post-transcriptional modification in eukaryotic cell, right? So capping, it is performed in presence of the enzyme guanosyl transferase. So C is also correct. Then last option you are given ambiguous codon. Okay, generally we know codons are non-ambiguous, but GUG, we know it's an ambiguous codon. How it is ambiguous? If it is present in the first position, initiating position, it will code for initiating amino acid, but other way it will code for valine. So this is how GUG comes under ambiguous codon. This is an exception, right? So all options are correct except the first one, catalytic RNA, okay, which cannot be the soluble RNA. So what will be your answer? It is first option A, right? Next, let's move to the question number 104. If egg of an organism has 10 picogram Pg of DNA in its nucleus, okay, what's your given? Egg. Egg is the gamete, always haploid. It's having 10 picogram of DNA, right? You are asked how much DNA would a diploid cell of same organism, diploid cell, okay, diploid cell will have how much? In the G2 phase of meiosis, G2 phase. So diploid cell, it will have double the amount, surely 20 Pg of DNA. And you are asked where? G2 phase. And we know that during the S phase, there will be duplication of amount of DNA. DNA duplication will occur, replication of DNA will occur. So amount of DNA, content of DNA will become double simply. So very after the S phase, that is in the G2 phase, now the amount of DNA will be double, right? So the answer will be what? 40 Pg for 104. Got it? Okay, fine. Now let's see next question, integumented megasporangium. It is the technical term for ovule. It is first developed in. So integumented megasporangium. You know this question was asked in NEET also, that what is the technical term for ovule? It is integumented megasporangium, right? You know this very well. Megasporangium, it is the structure inside which megaspores are formed. And what is integument? It is the protective covering. So integument with megasporangium together we call as ovule. And ovule, it was first evolved in gymnosperm. Okay, gymnosperm. They are the first group of plant kingdom showing the evolution of ovule. So this is very simple. Okay, integumented megasporangium. It is first developed in gymnosperm, right? Other option, angiosperm, no. Okay, more that is here the ovule is more and more advanced. Like you know, in case of angiosperm, most common type of ovule we get is anatropous. But in gymnosperm, simplest ovule is formed, primitive ovule, that we call as orthotropous, right? Pteridophyte, no, they do not have the ovule, and algae, no. So for question number 105, answer will be A, gymnosperm, integumented megasporangium. Got it? Then 106, which of the following is an example of subaerial modification of stem? Stem, it can show three type of modification. Underground, okay, underground stem modifications like you studied about rhizome, bulb, okay, corm and tuber. Another category is subaerial. In this subaerial stem modification, you studied about runners, stolon, offset as well as suckers. Then aerial stem modifications are like phylloclade, cladode and tendrils. So three categories of stem modifications are there, and here the question is on subaerial stem modifications. Fine. So let's see now for 106. Subaerial stem modification. Ginger, it's an example of what? Ginger is an example of rhizome. So that is an underground stem. Potato, tuber, underground stem. Jasmine, correct. Jasmine, it is a subaerial stem modification. Okay. Which subaerial stem modification? Stolon. Right, stolon. This is the bud which will sprout to form subaerial stem. Okay, this is the subaerial stem, stolon, which will grow horizontally, okay, and it will arc down. It will show arcing like this because it is longer, slender, fine. Obliquely it will grow and then it will arc down, and on touching the soil it will develop adventitious root, and this bud again sprout to form, sprout to give what? Leafy shoot. So this is stolon showing unlimited growth, fine. So jasmine is an example for stolon, subaerial stem modification. These examples are all from NCERT book, very, very important. Please go to these examples, as I always say in class. For example, no need to see other book, NCERT is enough. Alright, next asparagus is given. Asparagus, it belongs to a aerial stem. What is that aerial stem? Cladode, right? So for 106, answer will be what? Jasmine, that is option C. Fine. Next, in which of the following disorders affected individual will have 47 chromosome? So normally a human possesses 23 pairs, that is 46 chromosome. This is having an extra chromosome. Fine, it's a type of aneuploidy, right? It's a type of aneuploidy having extra chromosome, and this is what type of aneuploidy? Trisomy, because having 47, that is 2n normally, but here one extra chromosome is there. Find 2n plus 1, trisomy, 46 plus 1. So, which disorders? Turner syndrome. Turner syndrome, we know that the number of chromosome total is 45. Okay, that's a type of monosomic condition, Turner syndrome, a disorder, chromosomal disorders in female. Turner syndrome, the chromosomal constitution is 22 pairs autosome, that is 44, but having only one X chromosome instead of 2X. This is Turner syndrome. Then Klinefelter syndrome, another chromosomal disorders in male, Klinefelter syndrome. So, what is the chromosomal constitutions for Klinefelter syndrome? Autosome is 44, right, same, but having 3 sex chromosome instead of 2 sex chromosome, XXY, 1 X chromosome extra, fine. Then next is Down syndrome, which is another example of trisomy, but it is autosomal trisomy. Autosomal trisomy means having one extra copy of 21st chromosome, one extra copy of 21st chromosome. So, this is how the chromosome number becomes 47. So, 47 chromosome, it is present in Down syndrome as well as Klinefelter syndrome. So, what will be the answer? Last option, both B and C. So, questions are very easy, all from textbook, okay, NCERT book, direct questions. Now, let us see next question 108, incorrect statement you are supposed to find out. In dicotyledonous root, vascular cambium is completely secondary in origin, is it? Questions from anatomy, okay. In dicot stem, vascular cambium, it is completely secondary in origin. Yes, it is very true. Dicotyledonous root, oh sorry, it is not stem. Dicot, first option I am talking, dicot root, okay. Vascular cambium, it is completely secondary in origin. This is very true, because in dicot root, VC, vascular cambium is formed by the union of, fusion of first cambial strip and second cambial strip. So, VC, it is formed by fusion of first cambial strips and second cambial strips, right. So, this is completely secondary in origin. So, first option, it is right, you are supposed to find out incorrect. Second options, lenticels occur in mostly in older stem of woody stem. Yes, what are lenticel? Lamp-shaped area, lamp-shaped pore, mostly present in older stem, older tree, right, because it is secondary structures. lenticel woody plants में बहुत common है lenticels, meant for gaseous exchange as well as transpiration. So, ये woody plant, older plants में ये common है lenticel. So, ये वाला options भी right होगा, ठीक है. Then secondary growth occurs in stems of dicot and gymnosperm. Very true. Dicot and gymnosperm, they show secondary growth. So, C options, it's right, that is secondary growth. It is common in stems of dicot and gymnosperm. So, this is right. Last After secondary growth, primary medullary rays are more prominent in dicot root as compared to dicot stem. But students, it's wrong. As we know, in dicot root, pith is very negligible. Either you can say it is absent or very poorly developed. So, medullary rays are not seen in dicot root. So, this statement is wrong. It is incorrect. Medullary rays are more prominent in dicot stem, not in the dicot root. Got it? So, last option here wrong hai. So, answer will be what? This one. For 108, D option is the correct answer. Okay, let's see next questions. Question number 109. Cell organelle which is not involved in photorespiration. So, what is photorespiration? This is a wasteful process we consider, okay, which will decrease the photosynthetic efficiency, photorespirations. There will be oxidation of cyclic compound, oxidations of compound, photorespirations. This is similar to respiration in some point. There will be photorespiration is what? There will be extra release of oxygen, extra release of photorespirations, extra release of carbon dioxide and extra intake of oxygen, input of oxygens, photorespirations. So, this is an wasteful process, mostly it is the characteristics of C tree plant. But this is not respiration because the process can occur only in presence of light, okay, and it occurs only in green cell, only in plant. So, photorespiration involve 3 cell organelle. These are chloroplast, peroxisome and mitochondria. Okay, chloroplast, there will be oxygenation, then peroxisome, again oxygenation will occur. Oxygenation will occur, input of extra input of oxygen. Mitochondria, there will be release, it will show release of CO2, carbon dioxide. So, these 3 cell organelles are involved in performing photorespiration. Photorespiration occurs only under certain special conditions like high temperatures, high light intensity. Okay, the process can occur under certain special condition, high temperatures, high light intensity, okay, and when oxygen concentration is high in the atmosphere, the enzyme Rubisco, okay, enzyme Rubisco will show what? Oxygenase activity. Fine, oxygenation reaction will occur. So, very simple question, cell organelle which says not involved. So, what are involved already I have told and you studied, glyoxysome will be answer. Okay, glyoxysome does not play any role in photorespirations. Fine. So, very simple question, you have got all the simple questions. Hope you did not make any mistakes. Alright, so 109 ho gaya, next 110. Again very simple, chromosome having only one arm. So, straight way you can do it, this is telocentric. What is telocentric? That centromere is present at the terminal end. It appears that only one end of chromosome is there, telocentric. Position is terminal. Whose position? Centromere position, right. So, for 110, I know all you did correctly, this is C, telocentric. Fine. Which of the following is the reason for species diversification in tropical latitude? So species diversifications के लिए कौन सी statement correct होगा? Let's see. They have remained relatively undisturbed for millions of years. Very true. In the tropical areas, species remain undisturbed. So 111 की जो first option है वो right है. We will also see the other options. More solar energy is available, correct, in tropic, which contribute to lower productivity? No. More solar energy is available in the tropics, tropical area. So it will what? Increase the productivity, not lower productivity. So second option is not correct. They were subjected to frequent glaciations in the past. No. Tropical area remain undisturbed. A and B are correct. So B is not correct, already you have seen. So what will be the answer? That is first options. Most of the duplication of cell organelles occur in following phase of cell cycle. Such simple questions, students. Hope you have scored the full marks. Fine. Maximum cell organelle duplication occurs in which phase? This is the first phase of the interphase, that is G1, first growth phase. Okay, most of the cell organelle duplication will occur. So A option will be the correct, G1 phase. This is also named as energy phase. Why? Because ATP production will occur, right? ATP formation will occur, energy phase of the cell cycle. This is the G1 phase where the cell size will become bigger due to increased content of cytoplasm. Fine. So क्या होगा आंसर 112 के लिए maximum cell organelle duplication, that is A. Fine. Other options are not correct. So ये हो गया 112. Next 113. Doubling of chromosomal number in mitosis. Well, students, mitosis is equational division. That is, daughter cell, they will receive same chromosome number as that of parent cell. Daughter cell do not get double the chromosome as compared to parent. So this is which phase of the mitosis where doubling of chromosome will be there. So you remember that during the metaphase, okay, what will happen? Well, it's a centrosome, another centrosome. This is ester. Ester formation already done during prophase. Okay. In the metaphase, what did you study? That there is alignment of chromosome. Okay. Chromosomes become very, very distinct, right? Due to maximum condensations. So just simply I am showing this is alignment of the chromosome at the metaphasic plate, equatorial plate, and this alignment we call as what? Congregation. Now in the next phase, this is what? Metaphase. In the next phase of mitosis, which change will occur? There will be splition of centromere. Centromere will divide so that one part of chromosome moving to one pole, other part migrating to the opposite pole. So during the anaphase, what's going to happen? So during the anaphase, you know students that there will be splicing of centromere, okay, splicing of centromere, so that one part of chromosome, that you can say chromatid, okay, chromatid will move to the pole. There will be movement of chromatid, and in the movement always, you know, centromere will take the leading role, arms will trail behind. Fine. So this is what chromatid. But after reaching to the pole, it will behave like daughter chromosome. Fine. It will work like what? Daughter chromosome. Each daughter chromosome possesses only one double-stranded DNA, one chromatin fiber. This is we call as monad. Fine. So this is what, these are the events during anaphase of the mitosis, simply. So very after anaphase there will be nuclear division, that is telophase. During the telophase, nuclear division, karyokinesis will occur. Means this is going to be formed one nucleus, daughter nucleus. This will form another daughter nucleus just during telophase. Fine. So now what you can see, that in the karyokinesis, in the telophase, each daughter nucleus will receive equal number of chromosome as that of parent cell. See, here I am showing how many chromosome, only 2, means the parent cell will have 2 chromosome, parent cell is having 2 chromosome, right. In the anaphase, how many chromosomes are there in total? 4, okay, 4 chromosome in monad state. This 2 representing the chromosome of one daughter nucleus, this 2 will represents the chromosomes of another daughter nucleus. Means this is anaphase of the mitosis where chromosome doubling can be seen. Is it understood? Hope all you did correctly. The answer for 113, it will be anaphase of mitosis, right. Okay, let's see now next question 114. Which of the following statement is incorrect for simple diffusion? So it occurs along the concentration gradient. Yes, diffusion is the movement of particles of a matter always from the area of higher to lower concentration, follows the concentration gradient. So this is correct. It's a passive process. Yes. It is an active process. No. Energy is not required for the movement of the particle. So C statement is not correct. Last, it is not dependent on a living system. Yes, very true. Diffusion, movement of diffusing body, it is not dependent to a living system. So what will be the answer? So simple, it is C. For 114, the answer will be C. Fine. Okay, now let's see next questions. The question is on mineral nutrients. Which of the following is not true criteria for the essentiality of nutrients? As you know, there are many mineral elements present in the earth, different type, more than 105 types of mineral elements. But it does not mean that all the mineral elements are required by plant. They are essential. It is not essential. So which mineral elements are essential for the living, which are not? Scientists, they propose some criteria which we call as criteria for essentiality. So now let's see which is not the criteria of essentiality here. Number 1, element must be absolutely necessary for supporting normal growth. Yes. Means in absence of this element, the plant cannot complete its life cycle. It cannot show normal growth. So this is one of the criteria of essentiality. Then The requirement of the element must be specific and non-replaceable by another element, correct? Requirement of the element is very, very specific. It cannot be substituted by other element. Suppose say the plant has the deficiency of iron. So to meet the deficiency of iron, we cannot add zinc there. It is highly specific. So this is also true. Next, element must be directly involved in the metabolism of the plant. Correct. This is another essentiality criteria given by the scientists. Element must play a direct role in the metabolism of the plant. So next, deficiency of any one element can be made by supplying other element. No, it is not the criteria of essentiality. If the plant is suffering for a deficiency symptom, that deficiency symptom, it has to be met by adding that particular element only. We cannot add their other elements. It cannot be replaced, cannot be substituted. So the last statement is wrong, it's incorrect. So answer will be last option for 115. It is D. Okay, next. The isotope of carbon and oxygen used extensively for studies in photosynthesis. So isotope of carbon and oxygen, it is used, or which one? C14, it was used for the discovery of Kelvin cycle by the scientist Dr. Kelvin for tracing the pathway of C3, Kelvin cycle pathway. We know very well C14, it was given to chlorella, right? So that to know what is the fate of carbon dioxide in the fixation. So carbon 14 was used, right, for the discovery of the pathway of C3 cycle. Next, oxygen O18, it was utilized by scientists Rubin and Kamen to prove that the source of photosynthetic oxygen, it is water molecule, it is not carbon dioxide as thought before by the scientist. Okay, O18, it was utilized to discover, to know that the source of photosynthetic oxygen is water molecule. It is not the carbon dioxide, right? So what will be the answer? Second options for 116. C14 was utilized for the discovery of Kelvin cycle pathway, and the experimental organism was chlorella, was treated with C14. Then O18, it was utilized to prove that source of source of oxygen in photosynthesis is water molecule. Oxygen evolution occurs from water splitting only, and it was discovered by scientists Rubin and Kamen. And again they worked with same algae, unicellular algae chlorella. Fine. Chlorella, it was grown, it was allowed to grow on a medium where radioactive isotope of oxygen was used in water, and carbon dioxide, it was kept normally, and the medium was exposed to light. And after certain hours, what was seen that there was evolution of oxygen gas, and this oxygen gas which was tested, okay, which was tested, experimented, it was found to be radioactive. So in the oxygen of CO2, it was normal oxygen, but oxygen of water, it was radioactive, okay, it was radioactive isotope. It is the isotope of oxygen. So what does it prove? Oxygen, it evolves, it comes only from water molecule. Fine. So what will be the answer for 116? It is second options. Next students, question number 117. Ribulose 1,5-bisphosphate, it is present in which category of plant? Plant fixing C2 cycle or C3 cycle or C4 cycle? Yes students, you studied that Kelvin cycle, that is C3 cycle. It is the common cycle, it is the universal pathway shown by every category of photosynthetic plant. Okay, any photosynthetic plant, it will fix, okay, it will produce sugar. Sugar synthesis, it is performed only by Kelvin cycle. Kelvin cycle, it is the universal, it is the common pathway for sugar synthesis for any category of plant. And to perform Kelvin cycle, what is the carbon dioxide acceptor? It is RUBP, ribulose 1,5-bisphosphate. So ribulose 1,5-bisphosphate, it must be present in any type of photosynthetic plant. Okay, so what will be the answer? All photosynthetic plant for 117. Next, let us see 118, source of electrons. Okay, source of electron for ETS, electron transport chain during aerobic pathway. So, source of electron for ETS during the aerobic pathway, it will be both NADH as well as FADH2. Fine, electrons, it is both NADH as well as FADH2, because during the electron transport system, what we know that there will be oxidation of NADH. There will be oxidation of NADH by the complex 1. So, during the oxidation of NADH, there will be release of electron. Again, another coenzyme FADH2, it will be oxidized in the ETS by the complex 2, and due to oxidation of FADH2, again there will be release of electron. Fine. So, what is the source of electron in the ETS? Both NADH2 and FADH2, both can show oxidation to release electron. Fine. So, very simple, for 118, answer will be D, last one. Next, question number 119, how many carbon dioxide molecules are released during complete oxidation of one glucose molecule? By stepwise removal of all hydrogen atom in Krebs cycle of mitochondria. Yes. What you are asked, how many carbon dioxide molecules are released during Krebs cycle on the mitochondrial matrix for the complete oxidation of one molecule of glucose. Now students, we learned that before Krebs cycle link reaction occurs, and during the link reaction, one molecule of glucose, it is oxidatively decarboxylated into two molecule of acetyl coenzyme A. Okay, during the link reaction, one glucose molecule, it is oxidatively decarboxylated to form two molecules of acetyl coenzyme A. And this two molecule of acetyl coenzyme A, they will enter in the Krebs cycle for complete oxidation. Fine. Means there will be two turn of Krebs cycle. In one turn, one acetyl molecule of coenzyme will be burnt. Right. Means it will take consecutively two turn of Krebs cycle for complete oxidation of two molecule of acetyl coenzyme A. Now, during the one turn, in each turn of oxidation, 2 molecule of carbon dioxide will be released. Okay, 2 decarboxylation reactions are there. Fine, in each turn, 2 turns will be taken. In the first turn, 2 molecule of CO2 will be released, that is decarboxylations. Okay, decarboxylation reactions will occur, that is conversion of oxalosuccinic acid into alpha ketoglutaric acid. This is one decarboxylation, again another alpha ketoglutaric acid oxidatively decarboxylated to form succinyl coenzyme A. So, these are the 2 consecutive decarboxylation reaction occurs in each turn of Krebs cycle. So, 1 glucose means how many turn? 2 turn. So, how many CO2 will be released in total? 1 turn, 2 molecule of CO2. 2 turn means simply it is 4 molecule of carbon dioxide. Got it? So, what will be the answer? That is second option 4. Steps should be very clear to you. Any type of questions you are given from respiration, you can do this because it is completely conceptual. Once you learn the mechanism, you are never supposed to forget it. Okay? Any questions you are given, easily you can do it. So, answer क्या हो गया अभी 119 के लिए? सेकेंड बना ऑप्शंस. Okay, the question is, it is for the discovery of Auxin. From the diagram itself, we can understand. The first experiment of Auxin discovery, it was performed by Darwin. Okay, Charles Darwin and his son, by taking canary grass. Darwin took canary grass for his experiment. It is the first experiment on Auxin discovery. So, what Darwin did that canary grass, it was exposed to sunlight, unilateral light was given, and what did he find that after some time the tip of the coleoptile, it bent towards the source of the light. Okay, it bent towards the source of the light, right? Again in another set of experiment, what they did that, the tip of the coleoptile was cut, again exposed to unilateral light, and what did they find at that time, at that experimental setup, that there was no such bending or growth, no such curvature, as the tip was removed, coleoptile tip was removed from the canary grass. So, from this experiment they understood, they concluded that there must be some growth substance present at the tip of the coleoptile, okay, apex of the coleoptile, but they failed to isolate that particular substance, right. So, some type of growth substance is present at the coleoptile tip which is responsible for this type of growth. Finally, Auxin, it was isolated by scientist Wendt in 1928 while he experimented on oat seedling, that scientifically we called as Avena sativa. So, in the Wendt experiment, what he did, this is the Avena seedling, cut the coleoptile, right? And this coleoptile tip, it was placed in an agar block. Right? So that the substance, growth substance which is present at the tip of the coleoptile, it can diffuse on the agar block. Next, what he did, this is the cut coleoptile, this is the stem part without coleoptile. Okay, it is the stem without the coleoptile, and here in this stem which is without coleoptile, agar block was placed eccentrically, asymmetrically. See how it was placed, right? Agar block was placed asymmetrically. So what did he observe? That this agar block, okay, it was placed on that area. There will be what? Translocation of that growth substance. Okay, the side of the stem having the agar block, it will translocate that substance, but in other area, other side of the stem, right, it will not have that growth substance. How did he know that? Because what did he find? When the agar block was placed over the stem which was without coleoptile, what did he find? That there was bending of stem. Okay, stem bending was there, and stem bending was there to the opposite direction of the agar block, right? Stem bend opposite to the side where the agar block was kept. So what does it prove? That the side of the stem having agar block, here the auxin concentration is more. Okay, till that time it was not named yet. That substance concentration, chemical concentration is more, right? And the side where the agar block was not there, that chemical presence was, concentration was less. This is the reason why it will bend, okay, to the opposite side of the agar block. And finally that chemical was extracted, isolated, and for the first time it was named as auxin. Okay, auxin means to grow, right? Auxin, that is the growth. So this is the first growth hormone, first growth substance present, discovered in the plant, auxin, right? It's a very important experiment. So from the diagram itself, it is quite easily understood that the diagram depicts, it is the discovery of auxin secreted by coleoptile tip, right? So we will read this statement accordingly. Experiment used to demonstrate that tip of the coleoptile is the source of gibberellin. No. Experiment used to demonstrate that movement of auxin is acropetal. No, not showing the movement, acropetal. Experiment used to demonstrate that tip of the coleoptile is the source of auxin. Correct, tip. Without tip, there will not be growth. So C statement is right. And last option, it is irrelevant. Arrow indicates direction of air. No, it is the light actually. So for 120 question, answer will be C. So this is an experiment, okay, on auxin discovery. Let's see the next question, 121. One of the following not propagate through the modified stem, means it is not the example of modified stem. So let's see which one is not the stem. Ginger is a stem, you know, onion is a stem, bulb, colocasia, it is also a stem modification, that is corm. Sweet potato, correct. Sweet potato, it is a root. It's a root modification. What type of root modification? Tuberous. Okay, tuberous root, an adventitious root modification. So for 121, the answer will be D. So आप सबने किया ही होगा, definitely correct है. है ना, बहुत सिंपल क्वेश्चंस दिया हुआ है। नेक्स्ट, one of the most resistant organic material is seen associated with most resistant organic material. It is sporopollenin. Okay, sporopollenin, it is present in exine of the pollen which can form male gametophyte. Okay, sporopollenin, even it is the most resistant biological material too. No enzyme can degrade, can destroy sporopollenin. Fine. So sporopollenin associated with the male part of the plant, pollen, exine of the pollen. So what are the statement given in the question? Embryo, no. Then female gametophyte, no. Seed भी correct नहीं है, तो कौन सा correct होगा? Male gametophyte of angiosperm, yes, male gametophyte. So 122 के लिए क्या होगा आंसर? मेल गैमेटोफाइट, ठीक है। एंड क्या नाम है रेजिस्टेंट मटेरियल का? स्पोरोपोलेनिन, फॉर्म बाय ऑक्सीडेटिव पॉलीमराइजेशन ऑफ फैटी एसिड्स, स्पोरोपोलेनिन। फाइन। The type of pollination that brings genetically more diverse type of pollen grain to the stigma. So genetically more diverse type of pollen grain, okay. This is of course cross pollination. Okay, in the cross pollination genetic diversity can be seen. So cross pollination, another name is what? Allogamy or genogamy. So yes, this is the answer, genogamy. Fine, because different type of agencies are involved here. The most common agent for pollination is biotic. This is insect. Pollination by insect we call as entomophily. Other options क्या दिया हुआ है? Geitonogamy. So it's not diverse as compared to genogamy, because geitonogamy is what? Pollination within the same plant between different flowers, right? So genetically that's a type of self only, though physiologically it's a cross pollinations. Autogamy, self-pollination. Parthenogenesis, no. Formation of embryo from the egg without fertilization. So pollinations के साथ कोई भी relation नहीं है. So 123 के लिए क्या आंसर होगा? Genogamy A. Next question, the unequivocal proof that DNA is the genetic material in the virus, it came from the famous experiment of whose? It is Hershey and Martha Chase. Okay, we all studied this, it's very famous experiment, the first experiment, or you can say it is the unequivocal proof to prove that DNA is the genetic material in the bacteriophage. So this experiment, it was performed by Hershey and Chase by using bacteriophage, which were allowed to cause infection on the E. coli bacteria, right? So this is undoubtedly second option, Hershey and Chase. Right? Then question number 1 to 5, genotype of a husband and wife, questions on blood group. Husband का genotype क्या है? IA and IO. Wife का क्या है genotype? Female का IB, IO. So what you are supposed to find out, how many different type of genotype and phenotype are possible. So quite easy. So the alleles will be like this. One allele is IA, IO, and for the female the allele, different type of alleles will be IB and IO. So combinations will be what? IA, IO, 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 IB, ओहो मैंने क्लीन कर दिया. I was supposed to erase it. Okay, no problem. I will do it again. It's a new board now. Instead of cleaning, I have erased it. Okay. This is the genotype of male, right? This is the genotype of female. So देखिए एलील क्या होगा? IA, IO, and from the female these are the allele, right? So products easily you can find out. One combination will be IA, IB, one combination will be IA, IO, another will be IB, IO, and IO, IO. So these are the 4 different type of genotype. What are the phenotype? What are the blood group? This is AB, both are co-dominant. This is A is dominant over O, so phenotype will be A. B is dominant over O, B blood group, and this is what? O blood group. So how many genotype? 4. How many phenotype? 4. So what will be the answer? This is second option. 4 genotype and 4 phenotype. For the question 125, right? I know all you did correctly. No chance of doing mistakes. अच्छा, next में क्या है? Both the parents are carriers for thalassemia, one of the autosomal recessive disorders, है ना? Yes. What are the chances of pregnancy resulting in an affected child? So let's see this. Clean कर देती हूँ. Okay. Both the parents are carrier. So carrier means they must be heterozygous. Okay, I am simply showing like this. Both must be heterozygous. So what are the combinations of genotype in the child? These combinations may come. Fine. This is another combination. It is another combinations. Fine. 1:2, 2:1, 1:4, that is. So out of these 4 combinations, like this is the one combination out of 4 child, this is also another, and it is 2/4. So affected child will be which one? Affected, because it is an autosomal recessive disorder. Disease will come in the child only when homozygous recessive condition is present. So homozygous recessive condition is this. This child can be affected. It is the affected child for the disease thalassemia. So what is the percentage? 25%. Right? 25% chances are there for the pregnancy having affected child if both the parents are carrier, heterozygous. So answer will be 126 का हो गया B, second, 25%, right? Next, find out the correct match. A. Himgiri, you studied all this, it's given in your book. Variety of sugar cane, no. Himgiri, it's a variety of what? Wheat. Atlas 66, high protein content, wheat variety, yes. It's true, biofortified variety, Atlas 66. Golden rice, low content of iron, no. High content of iron and vitamin A rich. So it's wrong. Parbhani Kranti, variety of rice, no, it's a variety of windy. सो 127 का करेक्ट मैच कौन सा है? ओनली वन, दैट इज बी. फॉर क्वेश्चन नंबर 127 आंसर हो गया बी. ठीक है, एट लास्ट 66. Okay, so this question is given on the effect of UVB radiation. Which one of the following is not valid? Not valid, not the symptom for UVB. So देखते हैं पहला ऑप्शन क्या दिया है? High dose of UVB causes no blindness. Correct. Then it damages DNA and mutation में occur. Yes, ये भी सही है, आपने पढ़ा होगा, बुक में डायरेक्टली दिया हुआ है. Then it increases the melting of polar ice cap. No, this cannot be effect of UVB radiations, melting of ice. It causes aging of skin, yes, and damage to skin cell. Very true. So कौन सा आंसर रॉन्ग हो गया? कौन सा ऑप्शन रॉन्ग हो गया है? This is C. Statement C is wrong. So 128 के लिए क्या हो गया आपका आंसर? इनकरेक्ट स्टेटमेंट आपको फाइंड करने के लिए दिया है, ठीक है ना? सो हो गया सी ऑप्शंस, राइट. चूज ऑड वन आउट विथ रेस्पेक्ट टू न्यूक्लियर मेम्ब्रेन. ओके, थोड़ा पानी पी लेती हूँ. with respect to nuclear membrane, inner membrane is smooth. Yes, students, this is the nuclear membrane, let's say. So this is the nucleus, let's say, right? It is the nuclear matrix, that is nucleoplast. It is the nuclear membrane. ये हो गया outer membrane, outer nuclear membrane. This is ectokaryotheca. Inner nuclear membrane, this is एंडोकैरियोथिका. सो क्या दिया हुआ है? इनर मेम्ब्रेन इज स्मूथ. यस, इनर मेम्ब्रेन एंडोकैरियोथिका ये स्मूथ है, करेक्ट. सो फर्स्ट ऑप्शन इज राइट. ऑड वन आपको ढूंढने के लिए दिया है. साइटोप्लाज्मिक सरफेस ऑफ न्यूक्लियर मेम्ब्रेन में बी अ राइबोसोम. साइटोप्लाज्मिक सरफेस कौन सा है? दिस साइड, ओके, विच इज टुवर्ड्स द साइटोप्लाज्म. Yes, we know now, students, they may be a ribosome. Cytoplasmic surface, that is outer surface. Okay, it may be a ribosome. Which ribosome? It is ATS ribosome. So this is also true. Outer membrane bears 70S ribosome? No, it is ATS. Perinuclear space is 10 to 50 nanometer. Very, very true. This is perinuclear space, space between outer and inner nuclear membrane. And what is the measurements given in NCERT? 10 to 50 nanometer. So कौन सा options odd हो गया है? That is C वाला. Outer membrane possess 70S. No, 80S होगा. So 129 के लिए C हो गया आंसर. Then 130 next. Which of the following statement is best to describe a climax community? So climax community, it is the ultimate community of a ecological succession. the community which is complete harmony with the environment. It is the most stable community. We say climax, say like tree. It is the climax community of ecological succession. So trees, they are stable, they are highly diverse. Got it? So let's see what are the option. More stable and less diverse. No, stable तो है, but less diverse कैसे होगा? More diverse. More stable and more diverse. Yes, ये वाला options correct है. Less stable, no. Less stable and less diverse, no. So for 130, such a easy questions, that is more stable and more diverse, climax community. Next, when a sparrow eats seed, fruit, insect and worm, then it is a what? Sparrow eating seed, fruit, we can call as primary consumer. Okay, but eating insect, worm, what we will call? Secondary consumer, or you can say primary carnivore, है ना? So sparrow दोनों category में आएगा. Sparrow will occupy two different trophic level in the ecosystem. Primary consumer as well as secondary consumer. Primary consumer, or you can say primary carnivore. So दोनों होगा, A and B are correct. Occupying two different trophic level. So 131 के लिए कौन सा हो गया आंसर? That is last. Primary consumer, primary carnivore, both. Next, in Human Genome Project, the sequence of which chromosome was completed in May 2006? You know, sequencing is already completed in 2003. The only one chromosome which is completed in 2006 means more time is required. This is the chromosome number having highest number of gene. This is chromosome 1, right? So 132, this is chromosome 1, that is second options. So आप सबने पढ़ा ही होगा, ये एनसीईआरटी में डायरेक्ट दिया हुआ है. This is salient features of human genome projects. Okay, next is again from ecology. Eminent conservationists identified that maximum protection is required to biodiversity hotspot area. Hotspot means they are very rich in diversity. So which of the following statement is most appropriate, okay, for this? So we will read this statement. Hotspot biodiversity protections के लिए पहला क्या दिया हुआ है? These are the region with very high level of species richness, correct? Hotspot is what? Having maximum species diversity. So पहला statement तो right हो गया. Second, these are the area where alien species were introduced. No, no alien invasion in hotspot area. So second statement wrong है. These are the region with high degree of endemism, correct? ऐसा बहुत सारा species है जो सिर्फ उसी area में present होगा. Endemic to that area only, not present anywhere else. That's why they should be protected. So third statement is again correct. These have a history of religious and cultural tradition. No, not such. कोई ऐसा religious या cultural traditions नहीं है hotspot biodiversity में. So पहला and third ये दोनों statement right हो गया है for the protection in terms of hotspot area. So कौन सा ऑप्शन होगा? Yes, C वाला 1 and 3. So 133 के लिए क्या हो गया आंसर? C. Statement first and third दोनों करेक्ट है. No chance of making mistakes ना. Very easy questions. Next, questions on hemophilia, one of the X-linked recessive disorder. It is a genetic disease. So, which of the following is correct for hemophilia? It is an autosome linked dominant trait. No, neither autosomal nor dominant. Then affected male should have unaffected father. It is not correct. Affected male not. It should have diseased father only. Heterozygous female cannot transmit the disease to her son. So आपको क्या फाइंड करने के लिए दिया है? करेक्ट. सो पहला और सेकंड ये दोनों स्टेटमेंट रॉन्ग हो गया. Heterozygous female cannot transmit the disease to her son. No, it is possible. Okay, it will show what? That progynic transitions disease can be transmitted from heterozygous mother, carrier mother to her son. So ये भी wrong है. Then possibility of a female becoming a hemophilic is extremely rare. Yes, that's true. Okay, possibility of hemophilia disease, it is more in male. Okay, it's very rare in female, because in female that disease will appear, disease will occur only in homozygous conditions, but in male disease can be disease can be seen even in heterozygous, hemizygous conditions, right? Hemizygous conditions. Okay, so 134 हीमोफीलिया के लिए कौन सा ऑप्शन करेक्ट हो गया है? लास्ट. ओके, पॉसिबिलिटी ऑफ डिजीज इज वेरी लेस इन फीमेल. नेक्स्ट, फेलियर ऑफ सेग्रीगेशन ऑफ क्रोमेटिड मीन्स एनाफेज ड्यूरिंग सेल डिवीजन साइकिल रिजल्ट्स इन एन्यूप्लॉइडी. करेक्ट. Down syndrome result due to, so one such question already we have solved, aneuploidy. Okay, so this is a genetic disorders, you can say aneuploid, right? Aneuploidy. So here aneuploidy will arise only when there will be failure of disjunction. What is disjunction? It is separation or segregation of chromatid during the anaphase of the cell cycle. So if the chromatid or if the chromosome fail to segregate or fail to move towards the pole, the organism can show aneuploidy conditions. Gametes will be abnormal gamete. Okay, abnormal gamete. See like this. Yes, I am explaining for 135. Suppose say it's a normal cell, just I am saying, okay, having 8 chromosome in diploid conditions. So this is one homologous pair, second homologous pair, third homologous pair and fourth homologous pair. So during the anaphase what will happen? One chromosome of the pair move to one pole, other to the opposite pole. Same for the second pair also. ठीक है? Same for the third pair, one chromosome to this pole, other to opposite pole. Right? So what will happen? That gamete which is formed in this side, it will have how many chromosomes now? 4. Same, the gamete formed or the cell which is formed, gamete which will be formed, it will also have 4 chromosomes. This is what? Normal meiosis we say. This is disjunction, separation of the chromosome. But in case of aneuploidy, what will happen? Again I am showing 4 homologous pair. So from the first pair, one chromosome to one pole, other to the opposite. Same for the second and third. But now it can happen that this fourth homologous pair, instead of moving to two opposite pole, it can show movement to one pole only. Okay. There is no segregation of chromosome for the fourth pair, just for example. So what will happen? The gamete which is formed in this side, okay, it will have one chromosome less, that is 3 chromosome instead of 4. It should have n, but what it will have? N minus 1, 1 chromosome less. Gamete or cell which is formed in this side, it will have 1 chromosome more as compared to normal, 5 chromosome instead of 4. So these are what? Abnormal gamete. This is what we will call disjunction. So now मान लीजिए ये जो abnormal gamete है n-1, इसका fertilization हुआ with a normal gamete. So कैसा condition form होगा? 2n-1, मतलब 1 chromosome less than the normal. This is what we will call monosomy. Okay, ये monosomy है. और ये वाला जो gamete है, abnormal gamete, अगर इसका fertilization normal gamete से हो गया, then what will be the chromosomal complement? 2n plus 1, 1 chromosome extra. This is what? Trisomy. This is what? Monosomy. So what are these? These are aneuploid. So aneuploidy, it is formed due to failure of segregation of chromatid or chromosome. Okay, failure of segregation of the chromosome. Right? So क्या था question? Yes, Down syndrome. Down syndrome, you know, it is an example for trisomy 2n plus 1. So what may be the reason for Down syndrome? Gain of extra copy of chromosome 18, no. Gain of extra copy of chromosome 21, right? So 135 के लिए क्या हो गया आंसर? Second options, B options, right? Yes, students, now we have reached to section B. So all questions are not mandatory, but we will give the solutions for each and every questions. So let's start with question number 136. In the 5 kingdom systems of classification, Chlamydomonas and Chlorella, they are placed in kingdom Protista, right? So 136 के लिए आंसर क्या हो गया? A. ठीक है, Chlamydomonas, Chlorella, unicellular organism, all are placed in kingdom Protista. Next 137, the DNA strand showing replication using Okazaki fragment also shows Okazaki fragment, okay? Just a part. This is the replication fork, parental strand. So, using this template, this is the new DNA which is formed with anti-parallel polarity. Fine. wait, I am using some different color. Okay, it is another new DNA. So, two daughter DNA are formed. One daughter DNA is formed continuously, and this is continuous DNA we call as the leading strand. This is the leading strand, continuous, and this DNA you can see it is formed discontinuously with again opposite polarity. Okay, opposite polarity, and this DNA, this newly formed DNA you can see it is formed in segment, in fragment. This is discontinuous strand or lagging strand. And यहाँ पे जो मैंने येलो कलर से मार्क किया, this is primer. Okay, these are the primer, right? So ये जो segment है, DNA का segment है, this segment we call as what? Okazaki fragments. Okazaki fragment, right? This DNA segment we call as Okazaki fragment. So question क्या है? Using Okazaki fragment also shows continuous growth in 5 prime 3 prime direction. No, Okazaki fragment it is not continuous, discontinuous. Then discontinuous growth on 5 prime 3 prime paternal strand. Discontinuous तो है, paternal yes 5 prime 3 prime, correct. Second options may be correct. We will also go to the other one. Discontinuous strand on 3 prime 5 prime parental strand. No, parental strand क्या है? 5 prime 3 prime. Involvement of one primer only. So that's also wrong. You can see more than one primer is involved, always in discontinuous or lagging strand. So what will be the answer for question number 137? Discontinuous growth on 5 prime 3 prime parental strand. So 137 के लिए आंसर हो गया B. Fine. अच्छा, you see what is the polarity of leading strand? Okay, ये वाला leading strand है, this one polarity. Again, board takes many time. Okay, see polarity of leading strand. लीडिंग स्ट्रैंड की क्या पोलरिटी है? दिस इज 3 प्राइम 5 प्राइम. ओके, लीडिंग स्ट्रैंड 3 प्राइम 5 प्राइम. लैगिंग स्ट्रैंड जो है ये वाला लैगिंग स्ट्रैंड, इसका पोलरिटी हो गया 5 प्राइम 3 प्राइम लैगिंग स्ट्रैंड. फाइन, because whenever you are asked for polarity, this is always we see the template strand. Leading strand ये है, template is 3 prime 5 prime. Lagging strand is this, template is 5 prime 3 prime. So Okazaki fragment also shows You are asked for Okazaki fragment, ठीक है, Okazaki fragment. Discontinuous growth on 5 prime 3 prime parental strand. Okay, 5 prime 3 prime parental strand. Discontinuous growth on 3 prime 5 prime parental strand. So what will be the answers? As I gave, it is second options. Alright, so now let's see next one, 138. The cross section of a plant material, it is a questions on anatomy, shows the following features under microscope. So क्या features है? एक, number of xylem bundles are more than 6 with exact protoxylem. Okay students, number of xylem bundle or phloem bundle more than 6 with exact protoxylem. So जैसे ही आपको exact protoxylem बोला है, definitely it is the section of a root. Okay, exact condition, it is shown by root, and number of bundle is more than 6. It is the diagnostic features for monocot root. Okay. Second, well developed pith. Yes, characteristics of monocot root. So plant material should be what? These two characteristics features are enough to identify that the section belongs to what? Monocot root. So 138. What will be the answer? This is last one. Fine. 139. If each amino acid weighs 200 mass unit, what is the weight of the protein molecule synthesized from an mRNA having 603 bases? Okay, students, 603 bases means how many amino acid? Number of amino acid will be how many? It is, because one amino acid means 3 base. Number of amino acid will be 201. One amino acid measures 200. Okay, so total number of amino acid is 201. Simply will multiply with the weight of single amino acid. So it comes out to be approximately what? The value given 40,000. So 139 के लिए very simple questions. That is last options. Okay. Next 140. Reservoir of protein in chloroplast during light reaction of photosynthesis. So we studied that during the light reaction of photosynthesis, at the time of electron transport, one more function goes on, that is pumping of protein from the stroma to the lumen of the thylakoid. मतलब जैसे जैसे इलेक्ट्रॉन का ट्रांसपोर्ट होगा, there will be pumping of protein from stroma to the lumen of the thylakoid. Fine. So there will be accumulation of protein in which part of the chloroplast? Always on the lumen of thylakoid, right? So what is the answer? Reservoir of protein in chloroplast during photosynthesis is what? It is the lumen of thylakoid. So 140 के लिए क्या होगा answers? This is lumen of thylakoid. Second options, right? Let's see next question, 141. Mendel laws predict accurately the pattern of inheritance. So Mendel, he proposed 3 laws. First law, law of dominance. Second, law of segregation. And third, the law of independent assortment. And we know very well, out of these 3 laws, the universally accepted law is law of segregation. Because in the law of dominance, there are so many exceptions like incomplete dominance, co-dominance, multiple allelism. Even in the law of independent assortment, some exceptions are there. They are not universals, like linkage, right? But the most accepted, universally accepted, valid law is segregations. Fine. So accurate pattern of inheritance कौन सा conditions में follow होगा? Alleles of a characters are co-dominant. No, then it will be a phenomenon of what? Co-dominance. Alleles of a characters are incomplete dominant. So incomplete dominance होगा. They will be different from their parent. Fine. Alleles of a character show complete dominance. Yes. If they will show complete dominance, they will be similar to parental conditions. Okay, it will show accurate pattern of inheritance if they will show complete dominance. Given character is determined by more than one gene. No, one character, if it is determined by more than one gene, then we will call as, this is what? Polygenic inheritance. So for question number 141, Mendel law जो perfectly match होगा, this is alleles of a character showing complete dominance. Yes. Next आइए 142, one turn of tricarboxylic acid cycle. This is Krebs cycle. So what are the output of one turn of tricarboxylic acid cycle? If there will be oxidation of one molecule of glucose, there will be two turn of TCA. Okay, because in one turn only one molecule of acetyl coenzyme A will be oxidized. So there will be two turn definitely for complete oxidation of one glucose molecule. So in each turn, what are the output of TCA? Okay, for each turn, output will be what? Means, you know how many NADH, how many FADH, how many carbon dioxide, water is not given, no, okay, no need to count water. Carbon dioxide, and then how many GTP or ATP. So in each turn there will be formation of 3 molecule of NADH, 1 molecule of FADH2, 2 molecule of CO2, 1 turn के लिए 1 molecule of GTP. Fine, for each turn. So what will be the answer? This is A. 3 NADH, 1 FADH2, okay, 1 GTP and 2 molecule of carbon dioxide for one turn. For two turns simply the number will be double. So क्या हो गया आंसर आपका 142 के लिए? It is A. See, cycle should be very clear, mechanism should be very clear to you. As I said, these are the things, these are the chapters, topic which are you are not going to forget it ever. जस्ट आपको समझना है, ठीक है, आप प्रैक्टिस कीजिए. एनी क्वेश्चंस यू आर गिवन यू कैन डू इट. देयर वोंट बी एनी टाइप ऑफ कंफ्यूजन्स, राइट. सो 142 हो गया. नाउ 143. वेरी स्मॉल एनिमल्स आर रेयरली फाउंड इन पोलर रीजियंस. या पिक आउट द मेन रीजन इन सपोर्ट ऑफ दिस. करेक्ट. स्मॉलर इज द साइज ऑफ एनिमल, हायर विल बी सरफेस एरिया, more chances of heat loss, right. This is the reason why smaller animal generally do not present, that cannot survive in polar area, small sized animals. So क्या हो गया? They have a larger surface area. Yes, larger surface area, more heat loss will be there. ये सही है. और other option, they tend to lose body heat slowly. No, tend to lose body heat fast. They have a smaller surface area. No, smaller is the size of the body, higher will be surface area, not smaller. They can generate more body heat through metabolism by expanding much energy. This is also not appropriate for the statement given in the questions. So as per the question, the answer will be what? That is very first option A. 143 यहाँ पे लिख देते हैं, आंसर विल बी ए, ठीक है। Question number 144. Number of prothelial cell in the mature male gametophyte of angiosperm. You know, in the angiosperm, in the male gametophyte, prothelial cells are not formed. Prothelial cell, they are present in gymnosperm, say in pinus. Prothelial cells, they have already disappeared in angiosperm. So, very simple, answer क्या होगा? 0, ठीक है। So 144, no prothelial cell in male gametophyte. So 0 कहाँ पर है? लास्ट में, हाँ, ठीक है। Then in a primary transcript, only exons are non-functional. Say this is the primary transcript, okay. Right, it is primary transcript. So two components are there, coding sequences, non-coding sequences. This is the coding sequences we call as exon, and it is non-coding sequence, it is introns or junk DNA we can say. So here what is saying, only exons are non-functional, no. Only introns are non-functional, both exons and introns are functional, and both exons and introns are non-functional. So here what will be the answer for 145? That exons and intron both will be non-functional. Why? Otherwise we know that exon, it can code for a specific amino acid. But why they are non-functional? It can turn to be functional only after the splicing process. Means post-transcriptional modification is required for the functionality of the exon sequences, right? So for the primary transcript, whenever you are given primary transcript, both exons and introns will be in non-functional conditions. And very after removal, very after the splicing process, the exons will be functionals. So for 145, as per the questions, the answers will be last options, that is D. Well, next one, 146, duplication of centriole. So students, one more questions was given related to this. Maximum cell organelle duplication, it will occur in very first phase of the interphase, that is G1. But duplication of centrosome, duplication of centriole, it will occur in second phase, that is synthetic phase. So 146, centriole duplication will occur in S phase, invisible phase of the cell cycle. So the answer is B. Next, sickle cell anemia is not due to sickle cell anemia, it is an autosomal recessive disorders. It is not due to frameshift mutation. Yes, it's because of the frameshift mutations, transition, transversion, and another one, substitution of glutamic acid by valine. So here all the options are correct except second, means it is not due to transition. It's an example for what? Transversion, substitutions. So for 147, the answer is here B. Sickle cell anemia, it is not an example for transition. Here purine base not replaced by purine. Pyrimidine, it is replaced by purine bases. So 147, it is B. 148, by knowing sequence of nucleotide on messenger RNA, one can predict the amino acid sequence, correct? This is the sequence of, you know, mRNA, sequence of the bases on the mRNA, from where we can predict that what type of amino acid will be synthesized. This is exactly according to the sequences present on messenger RNA. Genetic code is degenerate. Yes, second statement be correct है. क्यों degeneracy होगा? Because number of codons are more, very high as compared to the number of amino acids. 64 codons are there, but they will code only for 20 amino acids. So both these statements are correct here. So as per the question for the 148, the answer will be what? Both statement 1 and 2 are true. This is second options. So question number 149, what are the factors following they will inhibit the decomposition process. So this is an ecological functions, decompositions. So they will inhibit decompositions. First, high temperatures and aerobiosis. So this will induce the decomposition process, but low temperatures and aerobiosis, this will definitely inhibit decomposition process. So the factors which will prevent decompositions, this is low temperatures and anaerobic conditions, right, low oxygen concentrations. 150, the membrane of RBC of human, protein and lipid content. So it's a direct questions from your book, percentage is given. This is 52% protein and approximately 40% of the lipid for the membrane, cell membranes of RBC. So for 150, the answer will be second options. So students, this is all about the solutions for ANTS 11. Hope it will help you a lot. Thank you very much.